Welcome to Longevity Papers. This is our weekly explainer, where we sort through the latest research to find the stuff that might actually move the needle in aging science. Let's get right into it.
So I've got to be frank with you. This quote, it really sums up this week perfectly. Most of what came out is, well, incremental. It's not a week of huge breakthroughs. And that's okay. Our job here is to be truth seekers, right? To find the real signal in all that noise and see what could actually change how we think about the mechanisms of aging.
So here's what's on deck. We'll start with this whole signal versus noise idea, then look at a new clue in caloric restriction, a really interesting twist on the gut-brain connection, and a paper on epigenetics and neuroinflammation. And then, of course, we'll wrap it all up with our final verdict for the week. All right, section 1, signal versus noise. You know, this theme is really the heart of what we do. Science is a marathon, not a sprint, and most of the work is just laying one brick at a time. This week is a perfect example of that. We're going to kick things off with what I think is the most compelling paper of the bunch. But to really get it, we need to put it in its proper historical context. Okay, so our first paper, it's all about a topic we all know well, caloric restriction. This is probably one of the most robust interventions we have in longevity science, right? It works like a charm in all sorts of animals. But how it works in humans, the nuts and bolts at the molecular level, that's still a bit of a black box. This paper from Yale tries to shed a little more light on that. So here are the details. The paper is out of the Yale School of Medicine. Now, it's a preprint on bioRxiv, and remember that's pronounced bioRxiv, so it hasn't gone through peer review just yet. But what really made us flag this one was the fact that it's based on human trial data. That always gets my attention. So here's the headline finding. In humans, if you put them on a 14% caloric restriction diet for 2 years, which is no small feat, by the way, you see a big drop in this protein called C3a. Now, C3a is part of the complement system, which is a piece of our innate immunity. This gives us a really nice clean link between CR and tamping down that chronic low-grade inflammation we call inflammaging. But, and this is a big but, we have to put this in context. This isn't some discovery that came out of nowhere. I mean, look at this timeline. We've known about CR in rats since the 1930s. And researchers like Weiss-Corey started connecting the complement system to aging way back in the 2000s. So what this new Yale paper does is connect those two dots. It specifically links CR to this one part of the complement pathway, C3a. It's a refinement, it's an important piece of the puzzle for sure, but it's not a whole new puzzle. And this is where it gets really interesting for us, for anyone thinking about therapies. This gives us a target. So the question becomes, can we just block C3a with a drug and get the same kind of health benefits as restricting calories for 2 years, without the whole not eating part? That's the billion-dollar question, isn't it? Answering that is what really matters next. Okay, let's move on to our second paper. This one dives into the gut-brain axis. And let's be real, this is a field that gets a ton of hype, right? So we have to be extra skeptical. But what's cool about this study is that it suggests the connection might go in a direction we don't usually talk about. So this one is from Northeast Normal University, and it's published, so it's gone through peer review. The paper is about this protein, TSP50, in neural stem cells and how it apparently connects back to the gut to regulate cognitive decline as we age. A bit of a mouthful, but stick with me. Okay, so here's the twist. Most of the time, we hear about how the gut influences the brain. This paper flips that script. It's suggesting that a protein in our brain stem cells, this TSP50, can actually change our gut microbes, and that in turn affects how our brain ages. It's a brain-to-gut story. Now that's a cool idea. But the big caveat here is that the whole microbiome field is notorious for correlation, not causation. It's really, really hard to prove that one thing causes the other. Plus, this TSP50 protein isn't new. We've known about it for a while. So we're left wondering, is this TSP50 thing a big deal? Is it a major pathway that's conserved across species and has a huge impact on our cognitive health? Or is it just another tiny piece of this incredibly complicated gut-brain puzzle with a really small effect? The answer to that question makes all the difference for whether it's a real therapeutic target or just, an interesting footnote. All right, on to our third and final paper for the week. This one hits on a really fundamental part of aging, our epigenetics. Specifically, it looks at what happens in the brain when our DNA packaging, our chromatin, starts to get sloppy. This research comes from a top-notch institution, Charité in Berlin. It's looking at how losing heterochromatin, that tightly packed, silent DNA, can kick off inflammation in the brain. So the mechanism they're proposing is pretty wild. As we get older, we lose some of the proteins that keep our DNA packed up tight.
When that happens, these ancient, dormant viruses buried in our genome, endogenous retroviruses, or ERVs, can wake up. The cell sees this, thinks it's under attack by a virus, and launches an immune response. Boom, you've got neuroinflammation. It's a fascinating link between epigenetics and innate immunity. But again, it's building on a lot of foundational work from labs like David Sinclair's over the last 10 years.
And here we are again, at the big question, causality. Is this breakdown of our DNA packaging a root cause of our brains getting older and not working as well? Or is it just a symptom, another biomarker that tells us, yep, this system is aging? It's the classic chicken and egg problem. And figuring that out is absolutely crucial if you want to find a target that actually works.
Okay, so let's put a bow on this. We've walked through 3 of the most notable papers from this week. What's the final verdict? What should we take away from all this?
Well, the reality of science looks a lot like this. The vast majority of it, maybe 95%, is incremental progress. It's good work. It's necessary work. It's scientists laying the groundwork brick by brick. And this week, this was a 95% week. The papers we talked about are cool, they're mechanistically interesting for sure, but are they game changers? No, they're not paradigm shifts.
So let's just boil it down to the essential questions we're left with. From Yale, can a C3A blocking pill really give us the benefits of CR? From Northeast Normal, is that TSP50 brain-to-gut link actually causal and a big enough deal to matter? And from Berlin, is heterochromate loss a driver of aging or just a marker along for the ride? Answering these is what separates a neat finding in a paper from a real-world therapy.
And that brings me to my final thought for you. You know, we've just spent all this time talking about what incremental science looks like. So it begs the question, what would a real leap forward look like? What would a paper and that rare 5% slice of the pie actually have to show us to completely change the game in longevity? That's the kind of paper we're always on the hunt for. And that's the standard we hold everything to.